So now we're moving on to phloem. Phloem sap moves from source to sink. It's a long-distance transport of sugars and other and water from sources, which will be mature leaves, storage organs. Mature leaves are storage organs like root tubers and such. Sinks will be young leaves, which haven't fully developed yet, so they still take up more carbon than they generate through photosynthesis, as well as fruits, roots, flowers, and when they first put that, first put those carbon stores into the storage organs, so the storage organs can be sinks or sources depending on their point in their development. So we have two kinds of phloem cells, and the main transport occurs in these sieve tube elements. They're fairly empty cells, so they don't have a lot of organelles. They don't have any vacuoles. They have these P proteins which allow them to clog up the sieve plates if there's any damage. They have all these little holes along the side of the sieve tube, and this is where the companion cells sit. So the companion cells sit right along the sieve cells, and they have all of the cellular machinery to direct and control the phloem. And that's what you can see here. Here are those sieve tube elements, basically empty, but then you have in the companion cells all right up against the cell membrane and cell wall where they can be most effective at controlling the sieve elements. They have heaps of organelles for doing their own physiology as well as the physiology of that's required for the sieve tube elements. So the way that these work, phloem works due to turgor again and bulk flow. So basically, near the source, there is a high concentration of sugars. And so what that does is it creates a pressure pulse that flows through the phloem. And so there's this front to this pressure pulse. So in the morning, the phloem hasn't been working too much. And then in the morning, the phloem, you start to get photosynthesis and increase in this concentration. And as you go through the day, this concentration pulse moves down the phloem. There are several ways that that companion cells can get the sugars into the phloem. The first is a symplastic mechanism using these ordinary companion cells. Basically, in the mesophyll, you have a high starch concentration because that's where photosynthesis is occurring. Then in the companion cells, you have an intermediate amount of intermediate concentration, and then in the sieve elements, a little less. And so you still have transport through the symplasm from high to low concentration. You can also have apoplastic mechanisms. So we have these transfer cells. You have, which have very few plasmodesmata to the mesophyll cells. And what they have these pumps which pump sucrose into the companion cell against its concentration gradient. Creates a very high concentration in the companion cell, which then pushes it down into the sieve elements. So first ones were symplastic, these ones are apoplastic. And then there's one third kind, these intermediary cells. What they do is they allow diffusion of the sucrose into the companion cells through relatively small plasmodesmata. And then those sucrose sucrose molecules are combined into oligosaccharides, so big long chains of sucrose. And those don't fit back through the plasmodesmata, and the only direction they can go is into the sieve element cells. And so that's how these intermediary cells concentrate these oligosaccharides instead of sucrose in the companion cell, and that's how they create the turgor and that pressure pulse going down the phloem. All right, so that was the end of the fundamental topics in plant physiology. Now we're moving on to the ecophysiology, or the interaction between physiology and environment, or environmental physiology. And we're starting with eddy covariance. Eddy covariance is the method that we use to apply the concepts of measuring photosynthesis and transpiration in plant leaves to entire ecosystems. So we have these sensors, very similar to the sensors that we have in a leaf gas exchange system, but we put them on top of a tower. Our measurements, and so we also need to be concerned about how representative our measurements are of that ecosystem. Essentially, we need to put those instruments as high above the canopy as we can. We usually try to go for 2 to 3 canopy heights. This one here is about 2 canopy heights. The reason for that is that the representativeness of our measurements depends on the distance between the top of the canopy and our measurements. And so that's what I'm referring to here. Large distance between canopy and measurement. So tall towers have a much longer fetch. They can see more of the ecosystem because they have a longer distance between the sensors, which are up on high, and the canopy. We get the same thing with a short canopy. So with a short canopy, that distance again increases, and so we see a large difference between the canopy and measurement for shorter canopies. What we measure with these things, we measure what is known as net ecosystem production. And from that, we can estimate soil and litter respiration. We can estimate plant respiration or autotrophic respiration, and ultimately estimate how much carbon is being taken up by that ecosystem before respiratory losses, as well as how much of that carbon remains after things like cyclone and fire. And I wanted to focus on productivity or photosynthesis. So this is basically photosynthesis by all of the photosynthetic organisms, the primary producers in the ecosystem. And why I wanted to bring this up is because of this temperature peak. In photosynthesis research for the entire, at least probably 100 years, but for quite a long time, it's been well known that photosynthesis peaks at some temperature optimum. It'll be different for different plants. It can even be different for the same plant at different times of its life. But what's most important is that there is some optimum below which photosynthesis starts to decline and above which some photosynthesis starts to decline. And as I said, leaves can acclimate and shift their temperature optimum.